Hello everyone. So today our topic is enzymes. So without any wasting any time, let's start. So basically, what are enzymes? So to understand enzymes, we first need to understand what is a catalyst. So catalyst is a substance or material that speeds up the reaction without being used up in the reaction. It lowers the activation energy of the reaction by providing an alternative path for the reaction to take place. So basically, catalyst is a substance which we add during a chemical reaction. So we add catalyst, which catalyst speeds up the reaction without being used up in the reaction. So which means catalyst is not inside the products, neither it is present in the uh, reactants. So where does the catalyst come from? So when the reactants start to react with each other, it is the time when we add catalyst. When catalyst is added, the catalyst also reacts with the reactant and speeds up the reaction, after which products are formed. But when products are formed, catalyst could be easily removed from the products to separate the products, so catalyst is not present in the products. You can understand it better from the diagram over here. So as you can see here, these are the reactants, and this is the original pathway. The blue one is the original pathway of the activation energy without catalyst. As you can see from the reactants, where the reactants is written to the blue line, it is the activation energy without catalyst. You can see it's pretty high, but when catalyst is added, the reaction energy reduces. You can see here the red path, it's a small one. So this is an alternative pathway. The red one is an alternative pathway that is provided by the catalyst. And as you can see, the red activation energy is lower than the blue one, so catalyst lowers activation energy and provided an alternative pathway. So catalysts are present inside human body, are known as enzymes or biological catalysts, or more commonly known as enzymes. Now enzymes, what are enzymes made of? Enzymes are made of purely of protein, and they catalyze metabolic reactions. Metabolic reactions are chemical reactions that take place inside the cells, which is also known as cells metabolism. So let's move to the next slide. So we need to understand how does an enzyme work. First of all, we need to know that enzyme acts on a substrate to break it into products. So enzymes acts on a substance. So the substance is called the substrate, and after the reaction is completed, the products are formed. So let's move on to the next point. So the manner in which enzymes work is known as the lock and key. The enzyme is the lock and substrate is the key. The enzyme is an active site on the surface that is complementary to the shape of the substrate. The substrate fits into the active site of the enzyme, where the enzyme acts on the substrate to break it down. Once the substrate is broken down by the enzymatic action is complete and products are formed. Now the enzyme is free to catalyze the next reaction. So we can understand this by this diagram over here, lock and key model. So as you can see here, then as you can see over here, red the red substance is the enzyme. The red thing you can see is the enzyme, and this green square and purple triangle together are the substrate. So you can see the shape of the substrate, and you can see the active site on the enzyme. The active site is complementary to the shape of the substrate. So the substrate comes in and it binds with the active site of the enzyme. So it forms the enzyme substrate complex. So the enzyme it starts acting on the substrate and breaks them apart to form the products. As you can see here, the square is separated and the triangle is separated and are in different colors. So this means the product are formed, and enzyme is left as it is. So this means that enzyme is free to catalyze another reaction. The shape of the active site of the enzyme is complementary to the shape of the substrate, due to which we say enzymes are specific in the reaction. As an enzyme can catalyze only one type of reaction due to the specific shape of the active site. So as you can see over here, the shape of the active site of enzyme is complementary to the shape of the substrate, which means that this enzyme can only act on this substrate. We can say this is an enzyme A and this is substrate A. So enzyme A can only act on substrate A. It cannot act on substrate B or substrate C on any other substrate. It is because the shape of the active site of the enzyme is complementary to the shape of the enzyme of the shape of active site of enzyme A is complementary to the shape of the uh, substrate A. So which we can say that enzymes are specific in the reaction, and they can catalyze only one type of reaction. We can say this is reaction one. So this enzyme A can only catalyze reaction one. So this means that this enzyme is specific in its reaction. Now the next topic is effect of temperature on enzymatic activity. Now we need to learn that how does temperature affect the enzymatic activity. So enzymes work best on their optimum temperature. If the temperature is raised beyond the optimum temperature, the enzymes lose shape of their active site and are no longer able to catalyze any reaction as a substrate no longer fits in the active site, and we say the enzyme is denatured. Now we need to understand them. When the temperature increases, the enzyme obviously is a protein and it contains kinetic energy. So when the temperature increases, the kinetic energy of the enzyme increases. So the rate of effective collision between enzyme and the substrate increases, due to which the shape of the active site of enzyme is changes because of many collisions, and we say that the active site of the enzyme is denatured. So, due to as the active side of the enzyme is destroyed, the substrate can no longer fit into the active side of the enzyme, and we said that the enzyme is denatured. So, when the temperature is decreased below the optimum temperature of the below the optimum temperature, the kinetic energy of the enzyme and substrate decreases, all due to which they move and collide less, and enzymatic activity slows down or stops. So, when the temperature decreases, the kinetic energy of the enzyme decreases, due to which the effective collision between enzyme and substrate also decreases. So, this means that the amount of reaction taking place between enzyme and the substrate is very less. So due to which very less products are formed, or even no products are formed if the substrate does not fit inside the enzyme. Enzymes present inside the human body works best at 37 degrees Celsius. Is the optimum temperature of the human body is 37 degrees Celsius. So as we all know that the optimum temperature of our human body is 37 degrees Celsius, or 96 to 97 degrees Celsius Fahrenheit. 97 to 90 degree Fahrenheit, I guess. So this is the optimum temperature of the body. So the enzymes present inside the human body work best at temperature of 37 degrees Celsius. As you can see here in this graph, this is the activity, this is the temperature. So you can see at the peak point of the graph is at 37 degrees Celsius. Okay, so and it's 100 enzyme is giving its 100% activity at 37 degrees Celsius. As the temperature goes beyond 37, 40, 42, 44, 46, if we assume, the activity of enzyme decreases, and it's a sharp decrease, sudden decrease. As you can see here, this is the optimum temperature, 37 degrees Celsius, after which the activity of the enzyme decreases as the enzymes are getting denatured, until it's at the point it stops. Now let's learn the effect of pH on enzymatic activity. Now the enzymes work best at like the temperature. Similarly, the enzyme work best on an optimum pH. The changes in pH level can cause denaturing of enzymes. Enzymes have a pH level according to the surrounding. For example, due to the presence of, as we all know, that dilute HCl is present inside our stomach. So there is an enzyme called pepsin in our stomach. So this enzyme pepsin works best at pH level 2. It is because that hydrochloric acid inside our body, as we all know, that all acids are below the level of pH 7. So hydrochloric acid is a very strong acid, but it's diluted in our stomach. So the pH is of that acid in our stomach is 2. So in that medium, the optimum pH of the enzyme pepsin would be pH 2. And similarly for other enzymes present inside our body, the pH level of their working is according to the medium they are present in. So these are some names of enzymes and substrates. Protease acts on protein to break them into amino acids. Lipase acts on lipids to break them into fatty acids and glycerol. So when lipase acts on lipids, it's broken down into three molecules of fatty acids and one molecule of glycerol. Amylase acts on starch to break it down into simple sugars. These are some questions related to enzyme. What is protease as a result of digestion by protease? As we have learned before, protease acts on protein to break them into amino acids. So the answer would be A, amino acid. Now the second question, which statement about enzymes is essential to log and key hypothesis? Enzyme molecules are catalysts. Enzyme molecules can be damaged by high pH values. Enzyme molecules can be damaged by high temperatures. Enzyme molecules each have a distinct shape. So, which statement about enzyme is essential to the lock and key hypothesis? So, we all know that enzyme molecular catalyst and it is not related to the lock and key hypothesis. So, similarly we'll just cut it out, and option B is related to pH. We'll cut it out too. Now we have to select between C and D. So, C is enzyme molecules can be damaged by higher temperatures, and D's enzyme molecules have a distinct shape. So, lock and key hypothesis is all about the distinct shape, the active site of the enzyme. So, the answer would be D. Now, the next question is, which graph shows an enzyme catalyzed reaction in the alimentary canal which varies with temperature? The answer would be C, because this is the temperature graph related to temperature. As you can see, 37 degrees Celsius is the peak point of the graph, and after that there are 13 degrees. So this would be the answer. So this would be all for today for the chapter of enzymes. I hope you understood it well. We'll meet in the next video, which would be related to the topic of biological molecules.